Now we're going to get down to how do we move things forward, and it's going to be much more, I guess, small print, but hopefully it will help set us up for the conversations that we have to have going forward. So I thought I'd start by really defining developmental and epileptic encephalopathies. Now I know that perhaps 50% of the audience was here last year, so you've heard it before, but it's a tough concept, and I continually find people don't quite get the essence of it, so I do think it's worth going over that again. And then I'm going to go into the natural history studies, the what, the why, and the how. I think I managed to get this almost onto here before. There we go. So an epileptic encephalopathy is where this story sort of started about the children with the severe epilepsies. And when I say children here, I mean adults as well. And I look after many adults with these disorders, so we don't want to leave the adults out. But where the story starts really is you have to have a lot of epileptic activity. And the definition of this group of disorders, the epileptic encephalopathy. Encephalopathy just means disorder of the brain, that's all it means. And it's where you have lots of epileptic activity. And here in this EEG you see a discharge every second in this child's brain. And that constant epileptic activity, that hammering of the brain, if you will, contributes to how the child doesn't learn and often makes their behaviour very challenging, as you all well know. And this is more than just if you look at the brain and you see a brain malformation. There's more than just that brain malformation. But most of our children, most of the parents here with children, have normal brains on imaging. So really when you think about the epileptic encephalopathy idea, I think of it as really three things that go together. Most, but not all, of the children have seizures, sort of 95%. You must have epileptic activity, and we call that epileptiform activity, but it's the same deal. And it needs to impact on development. If it hasn't affected your development, it's not truly an epileptic encephalopathy. So let's just take that apart. And with the seizures, most children will have very frequent seizures. In some of the epileptic encephalopathies, that's just for a period of time, and then the seizures are left behind. But mostly they're not. They usually have many types of seizures. In some of the epilepsies, they'll begin all of a sudden. So one that's famous for that is myoclonic atonic epilepsy, where the child will come along and be fine, normal development, and then kaboom, out come all these seizures, including drop attacks, which are very nasty. Or it may become more gradually, as is often the case in Dravet syndrome. In some of the epileptic encephalopathies, the child will only have one seizure type, which are maybe just absence with eyelid myoclonias, where they have constant seizures all the time. But that's just one example where they won't have multiple seizure types. And then there's this small, very rare disorder called Landau-Kleffner syndrome, where 70% of children have seizures, but 30% do not, and they still go backwards, and they still have lots of epileptic activity. So the epileptic activity, that comes in many shapes and sizes, but there are distinctive patterns. So probably the most famous one is called hypsarrhythmia, which is the one that you have when you have epileptic spasms or infantile spasms. But it's certainly not the case for many, well, for some of you it's the case, but for many of you it's not. Many of them have multifocal discharges, little areas of the brain having epileptic activity all the time, and that might be every 10 seconds, it might be every 1 second, it might be every minute. The timing may make a big difference. What age does it start? And the type of seizure that happens at that age. So some of the seizure types are a bit age dependent. So for example, infantile spasms typically begin at 6 months, and in some of the developmental and epileptic encephalopathies, the children go from focal seizures to spasms, very common with SYNGAP1, CDKL5, and then they may come out, they may continue having spasms, or they may go into other seizure types. And if a child's never had epileptic activity, they don't really have a true epileptic encephalopathy. Having said that, with Dravet syndrome, it's very common in the first few years of life to have normal EEGs, and they do have both a developmental and later a more obvious epileptic encephalopathy. The third part of the definition is the impact on development. And in some of the children, their development's quite normal, and then they plateau or regress. And in some of the children, they're never normal, and then they regress. And if, as in KCNQ2 land, seizures begin in the first week of life, you never know if they were normal because their seizures have begun at the same time. And then this regression, or the development can slow or it can go backwards, and that can be stepwise or it can be one bad time and never again. There are lots of ways that can happen. What triggers developmental slowing in these children? It's most commonly seizures. They'll have a long seizure or something, or a cluster of seizures, and the parents will say, oh no, my child's just lost some skills. But it may be other things. It may be infection. It may be immunisation sometimes. It may be brain swelling. So all sorts of things can go backwards. So this is the same comment, but in a picture. You have a child who has many seizures. This is a little girl with SCN2A encephalopathy, very frequent epileptic activity. When I met her, she was having a seizure every 5 minutes, and she lost her skills. She was not normal, but she's 21 months here, and she lost more of her skills. Why does this all matter? What's all this about? Well, it's about the fact that maybe there's something we can do about it. And if you have a structural abnormality, such as tuberous sclerosis, and you, in this brain, which is a cut through the brain here, you see lots of abnormalities of the brain called tubers, and with some pioneering work at the Royal Children's Hospital here in Melbourne, Simon Harvey and Virginia Meissner are showing if you take out the tuber that is the nasty one causing the seizures, you can actually resolve this process of epileptic encephalopathy. What about for us in the genetic world? Well, there are now, as you heard, many genes, but sometimes just by knowing which gene, and here I'm showing you SCN1A for Dravet syndrome, if you just stop the wrong drugs, which are Tegretol or Trileptal, and you start the right drugs, which might be topiramate or stiripentol, or one day soon, hopefully, fenfluramine, you can improve their development and their learning. So that's what this is all about. But with the last classification of the epilepsies, we took this another step forward and introduced the term developmental and epileptic encephalopathy. And this is to say many of these children have two things going on, one gene causing slow development and one same gene causing really bad epilepsy. And so you might have abnormal development, and then you have this process of the epileptic encephalopathy on top of the abnormal development, and that's where it's critical that you work with your doctors, because maybe there's something we can fix. It's not the magic cure that we've been hearing about, but it's about, maybe taking this drug away, as happens in Dravet syndrome, you stop the Trileptal, the child's seizures reduce. Just by doing that, understanding the disease, you can make a difference. And you as parents can be really empowered to help your doctors with that. And I think we'll move from this clunky term, and I can it because I'm behind it, but we can move from this clunky term to a gene name, encephalopathy, and I think a lot of you are already doing that. You're ahead of the world with doing that, but it's an important way of thinking about it. And a lot of these diseases have a range of comorbidities, so it's not just about the epilepsy, and I'm going to return to that a bit later. And this little boy in the picture, he has got KCNQ2, and you can see he's got very severe impairment at the age of 4, even though at 2 his seizures stopped. So it's not just about the seizures, it's about the gene that's affecting your development. So that's by way of background, and I'm sorry if those of you who heard it last year slightly differently have a sense of déjà vu, but maybe you can go out and educate the world about it. And what I'd like to now turn to is where next. And we're going to hear about the fantastic work from Steve's lab and people all around the world really addressing gene therapy, which is where we all want to head desperately. But that's a long haul and one that we need to work together with you guys. And a lot of the hype is around what's called natural history studies, and I thought I'd put this talk together for you to talk about the what, the why, and the how. What is this all about? And here you see it's about building a foundation, and here you see one of our most famous foundations, which now has come to a reality. And this our beautiful opera house, you can see here, it has many hues, and I think that's really important because we have hues around gait and mortality and speech and many things. It's not just the seizures. So let's start with the why, and there's a lot of hype around the world about natural history studies, and it's sort of a rebadging, but perhaps making a more scientific approach to how we look at understanding each and every disease that we're interested in. So it provides a foundation to understand the disease, the features of the disease, the comorbidity, and also the spectrum. I'm not sure how much you know about your child with your gene, or with their gene, I guess, but you don't know that that gene may cause a whole spectrum of diseases. And so SCN1A Dravet syndrome, I go back to that because that's the poster child, the first one really that was discovered, and 90% of children with that Dravet syndrome have one gene, whereas many of the other diseases have many genes. And so why that's a good example is that SCN1A can cause just a few febrile seizures, nothing else. You could be in the room here and have SCN1A, and different mutations, and we're trying to understand that in more detail, can give you Dravet syndrome. So just the gene isn't enough. We need a lot more than the gene, and we need clinical research to take us forward with that. You all want to know, what about my children in the long term, as Danny just said so eloquently. And we want to know the long-term outcome. What supports is your child going to need? Are there things that will come out a bit later in life? For example, we have a paper in press about PCDH19 girls, and we have found that women can develop psychosis. About a quarter of the women in our older group have developed psychosis. It's still small numbers, but that's a really important thing for a family to know. And what about the mortality? What is it, the chance that my child won't make 20? And as many of you know, in last year or the year before, I think we had a talk on sudden unexpected death in epilepsy. A ballpark figure for Dravet syndrome is 17% of children won't make their 20th birthday. So I think that's incredibly important as a parent to know that, because if it happens, then a little bit of your brain knows it was a possibility, and it's the worst thing you could possibly have happen in your life. But to know it's a possibility actually helps you to prepare if that awful thing were to happen. And then importantly, which drugs work and which don't, and we can already find that out if you find enough patients with a disease. And what about alternative therapies? Should I be using a ketogenic diet, I shouldn't really call that an alternative therapy, but would I use that for my child? And there are specific syndromes, epilepsy syndromes, where I would use that very early on. So what about all this? This is essential information for you families, but for us clinicians, but also for the wider community in order to support us and to know what we're going to need in the future. Now, I've mentioned the word comorbidities. All epilepsies can have comorbidities, and often do, even people with the mildest epilepsies. If you're very normal life and you have epilepsy and you start having tonic-clonic seizures, it completely mucks up your life as well. So just remember, this is something for all epilepsies. And comorbidities means other features, other problems. And these, in this group of patients, we know are often about learning, developmental delay, intellectual disability, or global delay in some of our children. But also really important are the psychiatric group. Autism spectrum disorder is about a third of the patients, I think, with the developmental and epileptic encephalopathies. Behavioural problems are familiar to many people in the room. But other things, what about mood disorders and psychosis and things like that? But we don't stop there. We've got to think about other issues such as speech. And Dr. Samantha Turner's over there. She's done some seminal work on speech in GRIN2A disorders and in Dravet syndrome. I think there's a huge amount we need to do around speech, around gait. And we're looking at gait in Dravet syndrome and trying to think about trials there. About gut problems. People haven't really begun to deal with that at all. And I've touched on the mortality issues around these diseases. So why else? Natural history studies is really setting the foundation so we are ready to go when trials are ready to go. And the trial process you'll hear more about from Steve and I think from some of the other speakers. And that trial process is long, years, tedious, incredibly important to do it well so that we're not doing something that will harm the child, but it is the promise of the cure that we all want. We need to make sure with the natural history studies set us up so that if you have a new therapy, we can check, does it really make a difference? Or, you know, is it all the hope that we bring to the table that's making a difference? Does it really change how my child was going to fare? Also, does a new therapy actually make my child sicker? I mean, it's quite possible it could make something worse. Maybe it'll make psychosis worse. Who knows? What other effects does it have for that child? And maybe it'll have positive effects. I mean, you know, cannabis is the one that everyone talks about every week, multiple times a day in my life. And, you know, the CBD we know is safe, and we now know it works in some of the severe epilepsies, but we don't know about the rest of the cannabis. You know, I hope it's doing no harm, and that's what we have to worry about as well. And does it change the natural history? And I'm going to share 2 examples of how trials have shown this beautifully with natural history studies. So why else do we want to have the natural history study? Well, I think it's an incredibly important framework for trial development to be able to go to our regulatory authorities such as the FDA and NIH in America or the TGA in Australia, so that we have a rigorous ability to judge, does it make a difference? Are these children different? So we need it, and the regulations around it are really important that we get it right in setting it up. We have to understand the relationship to other diseases that may relate to the same gene, but I also think that by having a natural history study that takes all comers, we can compare findings between different diseases, which could be really helpful and help us to recognise things that we may have missed otherwise, maybe because our numbers are too small for a specific disease. And importantly, it helps us with the science to understand the biology of these diseases and to work together as scientists to bring together new ideas and go, hey Steve, I've seen this makes a difference. Steve, can you figure out if your mouse does that? And then he goes, oh no, not another idea, Ingrid. So I wanted to share a couple of examples with you of why or how natural history studies have really had a huge impact. And we're really lucky to have Kate here in the audience. Some of you will remember last year I asked Sarah to pop her onto our panel because Kate had just come back from Rome. And the reason they'd gone to Rome is their beautiful 3-year-old Tom, who had language delay and that was all, and then he presented with seizures and regression. And he was shown to have an incredibly rare disease called CLN2, and just at the time that we made his diagnosis, they actually showed that a new drug was developed, which is a drug that replaces what's gone wrong. So here it's 2 bad copies of a gene. It's called recessive inheritance, complete bad luck, but that's the way it is. And his body is not making enough of this protein, which is an enzyme. So this company thought, we'll put some more protein in, we'll replace that protein. So it's not actually gene therapy, it's if you like, replacing what's missing. And at the time that Tom was diagnosed, this replacement called serlipinase alpha had just been identified in 24 children, I think, to have changed the natural history and stop the progression. And here you see the data, and here you see the natural history. Now, the developmental and epileptic encephalopathies are nasty diseases, there's no doubt, but this is a disease that usually kills you within 2 or 3 years. That's the history prior to this drug. So a very, very nasty disease. And you can see here the natural history, and you can see their drug. So this is the magic bullet that you're all sitting in this room wishing I could just magic up for you, and I can't. But that is what the long haul is about, the work that we want to do. And here you see Tom just going for a wander at the time around when we first met, and I love the fact Kate keeps me up to date, and really he looks pretty much the same now, and this is now, what, about 3 years? 3 years ago. But what I also wanted to bring to you is the end of what Steve and the other speakers are going to speak about. When you find this magic bullet, what happens then? Well, to keep Tom well costs $800,000 a year per patient, and families, you are key to this. Getting together in your gene-based groups, but also as getter, as we've heard, finding money for research to take this forward. It needs families to lobby government to be able to get these miracles, miracle cures. So thanks, Kate, for letting me talk about that. The second one I want to talk about, but just by way of introduction to, I think, what my colleagues will talk about, this is gene therapy, and this is where you develop a compound that actually changes the genes and how they work. I'm not going to go into the detail, but just to show, this is the one for spinal muscular atrophy. It also got approval from the government, I think, last year sometime, and you can see here are the patients that had the placebo, and here are the patients that had nusinersen, which is this drug for spinal muscular atrophy, also a disease which kills you very rapidly. So there's hope. We're actually now in the game, which we weren't before. Now, one of the problems for our team is that there are so many diseases. We're talking about more than 100 genes, I think, at about my last count, but I suspect it's even more. And one of the things I love about this is that you get parents like you are that change the world and develop different foundations or different parent groups to actually take things forward. And that is critical. But one of the issues that's arisen, and you can see this when you go around the world like I do to so many different meetings, that each group is trying to some degree, to reinvent the wheel. Because every parent group says, we want a family support group. I totally support that, as many of you know. They all say we need to know what's going on to find new drugs. We need to have this well documented. And so there's a certain amount of reinventing the wheel, and I want to try and help that process. So I wanted to say, but natural history studies aren't actually new, and they are just being more formalised. And this is when we first found the gene with my wonderful collaborator in Seattle, Heather Mefford. We found CHD2 and SYNGAP1, and we showed that they were genes for developmental and epileptic encephalopathies. And in this paper, we looked at 500 patients. I think we found 5 CHD2 kids and 6 Syngapians. That was before Danny and Danny's children were diagnosed. But this is where the story starts, with discovery science. But then you need bright young people, like these young, quite senior young medical students, and you need very inspiring parents. And what happened was Danielle walked in and said, I've done a Facebook survey with our group, and look at these graphs. And she pulled out all these graphs in typical Danielle style and said, this is what's happening and this is what's happening. And I said, that's amazing, but let's take it to the next level, and let's do it very medically soundly, collaborate with the world, as we've done. And we really looked at 57 patients with SYNGAP1. Now, I should say that we didn't include about 8 patients because they didn't really have SYNGAP1. They had a variant in the gene, but it wasn't the causative gene for their child's disorder. Some of those actually, I think, probably were, but they weren't confirmed scientifically well enough. I think 3 of them were and 4 of them weren't. So I just raise that to say that if it doesn't match with the presentation of the child and the finding, maybe you're not on the money. And this is just to show you one figure from that paper, but it just shows you that 96% of the children had intellectual disability, most moderate, some moderate to severe, some mild. 73% have behavioural problems and high pain thresholds. So some things were more Syngapian and some things were more more general. So what's our vision? Our vision with natural history studies is to try and help the system to move along at the highest scientific level. We know that there are many gene groups that are beginning to develop their own ones, and that's perfectly fine, that's great. Other new gene groups are just starting. When you find a gene, you find 6 children with it. In a few years' time, you have 57. And that's sort of, with the new genomic testing, it's exploding around the world, as you know, because the parents are contacting you. We want to design something that's not gene-specific, but is what I call gene-agnostic. Everyone can come. And we want to start here, because we have a great Australian context, we have very good collaboration, we have great younger doctors who are talking after I am, who I'm going to talk about in a moment, who are really dynamic and enthusiastic. But I want us to reach everywhere. I don't want anyone in Chile or in Taiwan to feel lonely. I want people to know that we're interested. So what is a natural history study? Well, some people say, is it the same as a disease registry or is it different? And the answer is they're quite different. A disease registry is much more simple. It's an identifier for the patient. And one of the things we have to worry about is the same patient being in several registries. So we have to work out some identifier so we don't report the same person 10 times. And maybe their gene variant, etc. A natural history study is much more looking at the disease process. So that's just to emphasise that when people say registry and natural history study, they're different. A natural history study follows a group of people over time who have a specific medical disease, and they collects information to understand how a disease or condition develops. And hopefully also it will incorporate outcome measures and biomarkers, and we heard from Dan about wanting desperately to get biomarkers. What are our goals? To understand the disease, to improve diagnosis and patient care, to support healthcare management and development, for clinical research, but most importantly, I think, development of therapies. And here you see this pyramid of a whole lot of factors that have to go together to get new treatments for patients, and natural history studies are central to that. What about the stakeholders? Well, there are a host of different stakeholders that are important here: clinicians, regulators, industry, government, patient advocacy groups, and of course the families themselves. And they all have input, and you've just heard how the Dannies went to government and got $2 million for genetic epilepsy research, and that's a really nice example of where we all have to work together. So how do we do this? Well, we need to think about the challenges. We often have a limited knowledge of disease progression. We may not know of adults with a certain disease, for example. We may have very limited number of patients, which is an issue with being in Australia, and why I want to be all-inclusive. And we have limited resources and time, and that's a huge issue, because to have a registry but not to have anyone to upload the data means that it doesn't happen. And I think that that is something we have to think about very carefully. We need to plan carefully for a comprehensive but efficient study. We have to have sensitive evaluations, and that's a big issue. We need to get ways of evaluating change at a much more granular level than we can at the moment. We need really good data collection and sophisticated design and biostatistics, and we need cost-effective programs. And here you can see the different processes one goes through to actually develop a natural history study, to plan it. We want to find out what we don't know. We want to think about what do we want to do going forward, but that we want to be flexible. We want to be able to be dynamic and change that. We want to design it very carefully, and that is, I think, a year of an IT professional's life, to be honest. We want to understand our evaluations. We want to collaborate, get good data collection, biobank and monitoring. So just a few points here. Design. We need to think about all these points on the slide. What number of patients? Well, I think the more the merrier, because by getting more you get much more of an understanding of the spectrum of the disease. We want to think about collaboration, and I think I don't need to convince anyone here that this is a team effort and we need to work together. And we want to think about data collection. And this is a big issue because families want to put in their data and we want to put in our data, and they're different levels. So what are some of the potential issues? Sorry, my computer's just chosen to freeze. Isn't that a wonderful thing? Why are you doing this in the show? Sorry, everyone. No, it's comprehensively about to die, I think. I think it's because my time's up and it's just decided to stop. So what are the potential issues? One I've alluded to already is that you may be thinking you have a gene and it's the wrong gene, as the variant is not the cause. So we need to make sure the gene data entry is right, and we have to have really good curation of that. The other thing is the diagnosis might be wrong, and I have a video. Actually, Milo, come here and see if you can save my last few slides. Sometimes children with movement disorders are diagnosed as epilepsy and they're not epilepsy. So if you put in the wrong data, you'll get out the wrong outcome. And I envisage a natural history study with a parent end and a doctor end, so we can see the information that's come out of there. I'm trying to think about what my last few slides show. I know one of them is a picture of Catherine Howell, who's a paediatric epileptologist from Melbourne. We've worked together now for several years, and Emma Palmer, who's a geneticist from Sydney. They're both going to be speaking to you. And it's not power, I don't think. It's just stuck. But they are both going to be central to this development, and we are going to try and get some grant at the end of the year that will hopefully formalise a network around Australia, and then hopefully we're very keen to work with people around the world. The other thing I wanted to say is that there are many other good groups like Ampeduri's in Boston and groups in London, etc., that we can work together with. And the natural history study that I envisage, we will collaborate in a de-identified way, and you will be asked to consent to participate, and then data can be shared. So all the KCNQ2 children around the world can be brought together. And I think you've almost saved me there, haven't you? I'll just check if there's anything lastly on my last few slides. I don't think that one there. Beautiful. No, don't worry about that. Oh, no, you do need to worry about that. Sorry, you can't see that. That would help. Sorry about that, and I'll find it if you like. I think I was almost about there. So one comment was around the family history issues, is that the family groups, that there has been an issue around the world, and many of you may not know this, and I use the US as an example because it's got such a large population, but there are, 3 competing SCN8A groups that developed. In SYNGAP, there have been 2 competing groups. In KCNQ2, there's been 2 competing groups. In SCN2A, there's been 2 competing groups. In SCN1A, there have been 3 competing groups in one country. So there is, I think it's fantastic that families get together, but I want our thing to be just one step removed from that, so we can work on getting the science right. And here's the picture of the 2 young stars that I'm working with, and these are the sorts of issues that I wanted to do. Design a system that's meticulous, dynamic and flexible, maintains confidentiality as critical, and a front end for you to upload data and a back end for us to upload data. A hub and spoke approach was suggested by Ampeduri yesterday. I think it's a really good idea. We all have our favourite genes. I fortunately have too many favourites. Who do we need? Well, Mari from my team put this slide together for me because this is what you need for a natural history study. And it's true, you need good ethics people, you need good legal people, you need good, to make sure you don't cross the confidentiality. What does that cost? That's 1 year's salary. So it's a bit depressing, really, because we've got $50,000 to start this process, and we're going to put in grants, but, this is what, to do this at the right level costs. So I just want I know what I want to do, just need, a bit of money to do it. So just to conclude our natural history study of the genetic epilepsies, it's for all families around the world who wish to participate, particularly Australia and New Zealand. It's going to be gene agnostic, so all genes are welcome. We're going to integrate with everyone who wants to integrate, and we want to be ready, trial ready on the global stage, so that when trials happen, the sorts of things Steve will talk about, we are ready to go. And then the last thing is an ad. Michaela is here. Stand up, Michaela. She is doing a little, she's right in the dark at the back there. She's a med student last year, wants to be a paediatrician, and she's got a survey. It will take 20 minutes of your life. She's got them to give to you. What are the things that make your life hard? And there's so much else going on. Thank you to those who've returned it. I couldn't do any of this without our unbelievable group, apart from Sam, who's the boss, the leader of the group. All the rest of the team, some of the rest of the team are here, and I'm really grateful to them for being so enthusiastic. And the final slide is to thank you all. Without this, we'd go nowhere. So thank you.
We have treats for you. What questions do we have for Ingrid? Yes?
Well, I just want to speak in support of advocacy studies from a parent point of view. My son Tom was on the slide, and when he was diagnosed 3 years ago, I, like many parents, was looking at clinical trials, Gov, etc. And I saw natural history studies, and my first impression as a parent was, well, how will that assist my child? I realise now that was a completely sort of selfish point of view and narrow-minded, because we went down a different path, but the drug my son was on, which has been incredibly successful, has recently gone through the life-saving drugs process. And in that context, I had to speak to the expert panel of the life-saving drugs process and to convince them that this drug was actually life-saving, which is the requirement to get funding. And if I had stood up and said, well, my 3-year-old who you saw on the slide can still walk and talk, it wouldn't have meant anything. But instead, there had been a natural history study for my child's condition, and it showed that by the time you were 6, you have 0 in terms of motor language score, which means no ability to talk and no voluntary movement. And with that natural history study, I could show the scientist my son, and that's only one example, but somebody who at 6 could still do these things. And that was essential. I really feel that without that real-life comparison against the data, if that data hadn't existed, there's no way the drug would have been put on the life-saving drugs program.
And I dare say rightly so. It went from anecdotal to being scientifically undergirded, and you could, in essence, prove your point.
It did.
Right. Any other comments or questions that we have? Yes, I'm seeing somebody right there.
Just again on the natural history studies, how do we as parents get proactive about encouraging our medical teams to include our children in collating this data?
I think, well, firstly, obviously, your energy you can put on your data, which is great, and then you can ask your doctors to help. The real problem in the system, and Dan tried to set something up like this in the US a couple of years ago, and it didn't work, was because the doctors don't have time to put it onto the system. And so, I mean, one of the things, as you all know, if you see me, is I recruit everyone, because I got research grants that fund my wonderful team's time to put the data on, and so the money that I suggested just to develop the natural history study would be the money that you need to keep going, and then it's not such a difficult process because you get the medical letters, etc. But there is sometimes a disparity between what families think is going on and what we think medically, and that's why I think it's important to know what's going in at what level.
Do we have any other questions? Yes, we have one over here. There we go. And is anybody else raising your hand? I'll just spot you now. Yes, I'll come to you next.
Hi. I have a question about the difference between the background encephalopathic changes and seizure activity in these conditions, specifically KCNQ2, because that's one I know about. And that KCNQ2 is a spectrum. We know that there's like benign familial seizures at the one end and then KCNQ2 encephalopathy at the other end. But it seems that some kids, from what I've seen, that have a better prognostic or better phenotype, have normal background activity and then have seizures in between, but they still don't have normal development, but yet they don't have background encephalopathy, and other kids have a lot more background in encephalopathy. So my question is, if I'm making any sense, how important is those background, like the hypsarrhythmia, the burst depression, those sort of background encephalopathy changes to the prognostic developmental outcome? Does that make sense?
Made great sense, thanks, Jenny. So I think in KCNQ2, well, let's just go back before KCNQ2. We know as paediatric neurologists, if you see a newborn baby and they've got a terrible EEG like burst suppression, your heart sinks because you know that this does not augur well for that child. So with KCNQ2, there are a number of different points that you put together and you know that that child's probably going to be in the encephalopathy group. And like an abnormal background is a help to that. Whereas we also know KCNQ2 babies with benign familial neonatal seizures, so it's very mild, normal outcome. They can have lots of seizures at the beginning and look pretty sick as well, but often between the seizures they look better. So we're trying to put together I guess, templates where you can put together what the child looks like, what the EEG's like, and what the mutation's like, and try and develop more of a prognosis, which is what you're asking, I think. Will my child be in that mild group, the moderate group, or the severe group? But it's more complicated than that, because it's, although we talk about one gene, we know that we all have a genetic background of another 19,000 genes, and that can affect. So that can affect how you do. And so, for example, in SCN1A, in Dravet syndrome, I've got, I don't know how many patients, probably several hundred. I've got one of normal intellect. Why is that one person of normal intellect? What's special about her? And I think it's because she's got other genes that's supporting that. So that was a long answer. I hope that helped.
And I wanted to come to you next, yes?
Hi, my name's Tracy. My daughter is Jessica. She's 21. She has a malformation of her CAC and A1A gene. We didn't get this diagnosis until she was 17, 18. So I'm finding it really, really hard to find any information in Australia. I'm just wondering if you can have any advice on how to do that.
I think with all of these genes, we have to think globally. So I'm not, I mean, it's good to find... I don't mean that you should pack up your family and go to the other side of the world. Definitely do not mean that. But I mean that you need to Google and look around the world for other families. I always encourage my families to get on the groups and then to take everything with a grain of salt, but take the bits that suit them. But in A1A, there's a lot going on. It's a gene with many different presentations, so I think one has to be a bit careful, because it's like many of these other genes now. We've just got a paper almost impressed on KCNQ3 where there's a milder presentation. Well, not the mildest we know about, but it wasn't thought to be nasty, but now it's got a moderately nasty presentation. So a lot of this is happening rapidly, and you said you knew nothing 4 years ago, and that's because the genetics has exploded, which has been super exciting to go from not having a clue, and in fact these children were thought to have acquired causes, something had gone wrong with their brains, to being able to solve at least 50%.
Yes, Chris?
Hi. It's a gene specific, but just in terms of the natural history studies, like in SCN2A, we're very lucky that Catherine Howell is working on a SCN2A natural history study at the minute, and I was talking to Anna Patori last night about the work that she hopes to do in America. Will you pull all of that into what you're talking about?
Yeah, that's my vision.
Yeah.
Catherine's work will be part of this,
Yeah.
and she's the SCN2A person. Yes, I'm hoping, and we're collaborating already with Anne on many studies.
And that would be the same with other genes.
That's my
Yeah.
vision.
Yeah.
All right, and I'm seeing another question just there. Wait one tick. We're going to get you a microphone. Anybody else while we're getting that one? Just yes, I'll come to you next.
Oh, no,
Hi, I'm Karen. It's just a grassroots question. I'm a parent of a almost 22-year-old child with Dravet. Grassroots, I'm not necessarily associated or involved in. I do the smorgasbord, pick and choose what I like. How can we help with the natural histories? How can we?
You can help so
get
much.
Involved?
You're the essence of it, really. So by just becoming part of our research program, and at the moment this natural history study in a formalised sense has to be developed with just $1 million. But the reality is that we can recruit you to our research program already. Sam and I, we've built our program over far too long, I won't say how long, 25 years plus, and we have, I think, 20,000 or something people on our database who have all been recruited and have been wonderful, and some are mild epilepsies and some are severe epilepsies, but I'm interested in everything, and I think it's really important to be all-inclusive. So we're very keen, in fact, not just happy, but keen to recruit anybody anywhere in the world.
So if you're recruiting so specifically, then what happens? Does she...
Well, you're happy to be sitting next to Amy right at this moment.
There we go.
And
Done.
Amy is our boss.
There we
She's
go.
the
So
Chief
for
Research
other people,
Assistant.
Amy, can you raise your hand? If anybody wants to be recruited and give,
Well, we have
we're agnostic?
several of our people. Do you want to stand
Yes,
up, our
yes.
team?
If we can have
The
Ingrid's team
RAs.
stand up. So
We've got a few of them who've kindly given up their Saturday.
All
they're at
right.
the front desk.
So
okay.
yes, yes.
Oh, live stream people. Look up the Epilepsy Research Centre. Is there a
Epilepsy Research Centre.
Is there a URL? That's it. Epilepsy Research Centre in Melbourne, Australia, and that will show you some links. Thank you.
Yes, I've got a question right there. Yes, yes.
Hi, my name is Rachel. My son is 8 and his name is Jack. Within weeks of his birth, we have met you before once. He was about 8 months old. He was diagnosed with Ohtahara syndrome, so you were saying burst depression and your heart sinks, and he's one of those. Worst case scenario, everything is really bad. So we get this through your research, this letter to say he has SCN2A, but I was in labour when the lady called me in early stages, and I'm like, just tell me if it's de novo. And she goes, no, it's de novo. So like, okay, I can continue to have this baby and everything's fine. So I rock up to my doctors with this letter from your thingy, and I said, okay, what does this mean? What do we do now? And he said nothing. It means nothing. You've got a diagnosis of Ohtahara syndrome. We don't need to worry about anything else. You just carry on as it is. Today's been I'm actually really upset because I listened to that and we failed my son because we listened to that, and I met Chris and I'm listening to all these people and we failed our son because we listened to him and we've done nothing. And should we now focus on things specific to SCN2A or focus on the worst case scenario? Like, he was supposed to have passed away within weeks of his birth.
Gosh, I'm so sorry to hear that. I don't think you need to, bash yourself up. I can't think of a nice way of saying that, because we haven't got a treatment, so nothing has changed yet. But this GETA group is about how can we move this along, and it is moving things along. But at the moment, there isn't a magic treatment for SCN2A. I have to say it's a major focus of Steve's work. I don't think you're talking about that today, are you? It's major work of Catherine, Steve, Sam, me. We're really focusing on that. And in fact, but at the moment you don't need to, you haven't missed the boat in terms of what's out there. There's nothing out there at the moment. But that's what we're about. And I'm conscious that this is this meeting gives you a lot of hope, and yet you're going home to a very sick child, and we're not changing that today or tomorrow, but we are on a mission, and that's where we're all grouped together.
And that's where the journey is, and that's where the Odyssey
So there's
is.
nothing that we could have done differently
No.
4.5 years ago?
Nothing you could have done differently.
You know what, and can I just say, we all come when we come, and we're so glad you came. We're really, really glad you're here. You're here among friends, and I bet there's not another parent in this room who hasn't just had a moment of really giving themselves a hard time for something they did do or didn't think of. So you're here now. We're really excited that you're here. Connect to lots of parents as well as the scientists, because it's a journey. We need each other. We need everybody in this room. That's how we're going to get through it. We're going to do it together. I'm conscious that we need to feed you. I'm going to give one more person a chance to ask a question, then we're going to have Kelly Cloesy speak. But yes, I promised you a question. You will get morning tea, I promise.
Just a quick one. Hi Ingrid, my name's Renee. This is Damien. He's 2 years old and has KCNA2 epileptic encephalopathy. We haven't had the pleasure of meeting you yet, but we are in really good hands with Kat Howell and Andrew Kornberg, so we've been really well looked after. Just on that, we've got a really awesome Facebook group of KCNA2 children around the world. There's about 30 families, 3 different variants. Now, we don't have a formal foundation, and we do have some awesome fundraisers, sort of just doing things locally. Is there something we can do in the meantime to help our group prior to actually going into something like this? Would it help to have a foundation established, some more money behind us that we could help potentially?
I think there are 2 answers to that. I think that your group could easily be part of this. I do think it is good to get your own badging for yourselves in a way, because then you go along and you can ask a scientist specifically to work on that, or what's even better. I sit on the Drave Research Foundation Scientific Committee in the US. I think I'm the only non-US person on that committee, maybe there's one other. And they are a group of amazing parents, like our amazing parents, and they give out like $1 million a year of research for SCN1A. So I think in a way there is a reason for both.
Right.
But
do you
I
have a
think,
target number that we should aim for?
oh no, the sky's the limit.
Okay, right.
And best
one
wishes.
researcher?
Yeah,
Is there any around?
Yeah, and we'd
Cash
love
payment?
some.
No worries. Thank you.